Informed reading of a real-world evidence. When seeking to interpret real-world evidence, it is important to verify that it was properly designed, conducted, or reported. Here is a small, non-exhaustive checklist of elements to consider in order to assess the confidence you can place in the results of the study. As the French Health Authority indicates in its methodological guide concerning the design of the study, the first step is to write a study protocol, which must detail the justification and objective of the study, the hypotheses being tested for comparative studies, the data sources, the source and analyzed populations, the analysis methods, the conduct of the study and quality control, the validity and expected limitations of the study, and ethical aspects. Any amendments to the protocol occurring during the course of the study must be declared duly justified and listed. For the drafting and validation of the protocol, it is recommended to set up a qualified and multidisciplinary scientific committee. All these elements must also be mentioned in a transparent manner in the report or article. We will go through the different aspects listed. The source of data must allow the main objective to be met. It has been identified from the set of possible sources or has been generated specifically to answer the question. The limitations of the data source are stated in the discussion part, for example. They may be related to the collection, to the quality of the data collected, or to missing data, for example. Moreover, missing data and patients lost to follow-up are mentioned in the study. Concerning the source population, the cohort analyzed is defined in accordance with selection criteria. The date of entry and the date of end of follow-up in the cohort are defined. The population is representative of reality, that is, patients using the health product in current practice or having the pathology of interest, with a good representativeness of the centers and patients included. If it is not the case, potential selection biases are discussed and, if possible, quantified using sensitivity analysis. Concerning the analysis methods, the exposure to the health product is defined, as well as the way it is measured. The timing of the exposure to the health product and the collection of the data of interest is compatible with the chronology of the study. The endpoints are defined and measured. They answer to the research question. In the context of comparative studies, statistical methods have been put in place to minimize bias and reduce confounding factors, for example, adjustment, matching, weighting methods, etc. The confounding factors taken into account are justified. Sensitivity analyses are planned and performed to assess the robustness of the results. The external validity of the results are discussed. Are the hypotheses consistent with previously published studies that show that the link is possible? In addition, methodological limitations and possible biases are discussed and presented. This may be related to the different points mentioned above. In particular, the limitations of the data source do not have a major impact on the interpretation of the results. They are robust according to the different sensitivity analyses. That's it. You are now ready to read a study publication in real-world settings in an informed way